All right, class. This week in lab, we're going to cover the microscopic anatomy and organization of skeletal muscle. As with most weeks, we'll start off with a quiz, and then we'll move on to something of a two-part lab. So I'll have microscopic anatomy of skeletal muscle, which will deal with part one of your workbook for the week, as well as a look at the microscope slides to take another look at the muscle we would have looked at in the second week of lab. We're also going to move on and look at the physiology of skeletal muscle. For this, we'll use a computer simulation using the PhysioEx software, and the workbook part 2 is going to deal with that. So you should have gotten this CD packaged with your lab manual, so hopefully that looks familiar to you. Be sure to bring that to lab with you. We'll be using that with the computers in lab or with your own computers to go through that second part, the physiology of skeletal muscle. Now, a couple things to keep in mind is that when we use the term muscle, we could be talking about a few things here. First, we could be talking about a tissue. So this week, we're specifically dealing with skeletal muscle, but it could also refer to cardiac or smooth muscle as well. Additionally, muscle can refer to an organ, so like the biceps brachii muscle or any of the muscles we've already learned. And finally, we might be referring to a muscle fiber, which is simply a single cell of muscle tissue. So be careful when we're using the term that you're certain which phrasing we're referring to. And as you'll recall, the muscle tissue types are shown here. We're specifically going to be looking at skeletal muscle, and hopefully you remember being able to recognize that by its very neat parallel arrangement and the very obvious striations you see there. Here we're taking a look at the muscle as an organ, and obviously the muscle is going to consist of many muscle fibers. So the single muscle cell, also known as the muscle fiber, is just one of many muscle cells in that muscle organ. But also look to see that these muscle fibers make up this smaller component called a fascicle. So there are somewhere on the order of 10 to 100 fibers in each fascicle, and there's many of those in each muscle organ. There's also blood vessels that are going to be running through the length of this muscle organ, as well as a good amount of connective tissue protecting, covering, and subdividing that organ. And what we see here is on the outer layer of the organ, we'll have the epimysium, which is a dense connective tissue that covers the entire muscle organ. On a smaller level, we have perimysium, which is still connective tissue, but it's going to be subdividing the muscle organ and surrounding each muscle fascicle. And then down at the cellular level, endomysium surrounds each muscle fiber, further subdividing the fascicles themselves. And this connective tissue plays a role in separating these components of the muscle, also playing a protective role. But further, it's continuous with the tendons, so it acts as this connection to the tendon, as you see here, which allows the muscle to transmit its force to the skeleton. So here in this slide, you can see the epimysium is this coating, kind of iridescent, almost translucent coating covering the muscle organ. Perimysium is the coating surrounding the fascicles. And if you look here, you'll see that there's a multitude of fascicles here, each of them surrounded by perimysium. And then endomysium is surrounding the single muscle fibers, the single muscle cells. So the contractile element is a hugely important portion of the skeletal muscle, but there's also quite a lot of connective tissue we'll find as well. And in the histology slide here, you'll see the microscopic view of the muscle fibers. You can see them round, so it's a cross-section here, as well as each of the layers of connective tissue. Here we've now zoomed in on a view of the muscle fiber. So this is just the single muscle cell here. And you can see that there are many myofibrils further inside of the muscle fiber. So a couple things to look at. You'll see the nuclei. So we've got one right here. There's another here, another at the bottom of the screen there. So there are several nuclei in every muscle fiber, which is something of a unique property of skeletal muscle. Also, you can see that they're pushed up right up against the sarcolemma, or the cell membrane there. Basically, we've pushed everything, kind of crunched everything out of the way so we can fit as many of those contractile myofibrils in the cell as possible. You can see there are still mitochondria that are spread out throughout, kind of squeezed in between the myofibrils. They're obviously creating ATP, so they got to be in rather close proximity so those myofibrils can make use of that energy. Now let's take a moment to talk about some of the organelles of skeletal muscle. We mentioned the nucleus, which we can't see in this close-up here. You do still see the mitochondria kind of squished in between the myofibrils. And granted, we're trying to make as much room for contractile fibers as we can. So everything is kind of squished out of the way. That said, there's still some really important organelles other than the contractile fibers, specifically the three parts of the triad here. So we have the T-tubule, or transverse tubule, the whitish portion here that runs across these myofibrils, kind of cross-sectionally. And that's going to be connected to the terminal cistern of the sarcoplasmic reticulum. So this whole process is part of the sarcoplasmic reticulum, which is a specialized endoplasmic reticulum in the skeletal muscle cell. The biggest job it has here is going to store calcium. So we've talked about in class how calcium can be used as a second messenger. Calcium is not found intracellularly. So in this case, the sarcoplasmic reticulum has the job of withholding calcium from the cytosol. We want to keep it out of that intercellular fluid. So we use this triad structure to do that. Now we'll talk more about calcium in a bit here, but remember that the star of the show is the contractile fibers, but we have some other really important parts of the muscle cell too. Without those, it wouldn't be able to do its job. So we want to talk about the sarcomere. It's the functional unit of muscle. Basically, it's the smallest component that's capable of contraction. And we've got several really important proteins to deal with. Actin, tropomyosin, and troponin are going to be microfilaments. These are going to be important, basically cytoskeleton elements that allow for contraction to take place. And we also have myosin, which is a motor protein. It does the work, it does the pulling. And they're also going to be what contribute to the striations of skeletal muscle. So you can see here, the red pieces are the myosin proteins. The blue pieces are the thin actin filaments. And where we have the myosin proteins, it forms a darker band, a lighter band where there's no myosin, causing these striations you see across the skeletal muscle. With a closer look here, you see that the myosin proteins, again, those are our motor proteins in red here. They have all these little round projections. Those are going to be referred to as the myosin heads. They are the parts that are actually going to grab on to the actin filaments. And when they grab on, they're going to pull the actin pieces towards each other. So it's all going to be pulled towards this M line. And the myosin proteins are working to shorten the distance between these two Z discs that act as anchors for these proteins. So if we shorten this whole component, we can do this millions of times as all of the sarcomeres in the muscle do this and cause a shortening of that muscle organ. Now we mentioned a couple other microfilaments, so we still want to talk about troponin and tropomyosin. So here we're looking at a close-up of the actin filament. Up top here, the actin filament has changed colors and now to a reddish pinkish color. And the issue with actin is that it has these specific binding sites for myosin. So those myosin heads can't just grab onto the actin filament at any given place. They're a protein. They act in a similar way to a receptor or to an enzyme where they need a specific place to interact. And that specific place happens to be right under where this tropomyosin is lying. So the tropomyosin is this green-looking rope structure, and basically the yellowish part of the actin is where myosin can bind. So we have a problem. Tropomyosin is blocking the myosin binding site on actin. So if tropomyosin stays in that position, we cannot have contraction. Luckily, we also have this troponin protein, which acts to facilitate the movement and the freeing up of that binding site. And troponin is going to react to calcium. So as calcium does get released into the cytosol, it's going to bind to troponin, it's going to activate it, which is going to cause a movement that drags this tropomyosin filament off of the binding site. So hopefully you recall from chapter 6 when we talked very generally about second messengers causing a cellular response. In this case, calcium is one of our specific second messengers. And one of these responses is to activate this troponin protein and make that move off of, move the tropomyosin off of the actin binding site and allow for that contraction to occur. Now let's take a few moments and look at the bigger picture here. Now we all know that skeletal muscle doesn't act on its own. It needs some sort of command from the nervous system, and we're going to see that at the neuromuscular junction. Basically, the muscle is going to be commanded to contract by a motor neuron. So that motor neuron is going to interface at specific places on the skeletal muscle to send that command. And this motor neuron is not necessarily going to be commanding just a single muscle fiber. It's going to be innervating several muscle fibers, so they'll all work together. And the way this works, we know that neurons send an all-or-none signal, and at the end of that axon, even if it branches, the same signal is going to all of the different destinations. So we have all of those muscle fibers all working in the same way. And this motor neuron and all the muscle fibers that are associated to it are referred to as a motor unit. They're all going to work together. So a single command from the neuron is going to control all of those associated muscle fibers. And what we see is a specific neuromuscular junction. So this is the synaptic terminal of our motor neuron, and it's going to synapse not with another neuron here, but in this case with a specific portion of the muscle fiber. So here we have neurotransmitter being released, acetylcholine here. As the acetylcholine comes across the synapse, it's going to find acetylcholine receptors on the muscle fiber. And that's going to start the signal transduction, which is going to result in a release of calcium inside the muscle and allow for contraction. And here we can, again, take a look at histology, take a look at a closer look of how this looks under the microscope. And here you can see the very neat, organized, parallel skeletal muscle fibers. And here you can see the terminal of the axon. So right in this place here, we'll have a neuromuscular junction there. That axon will release acetylcholine, and that'll command a muscle contraction in that fiber and any other associated fibers. So here, this one is also from the same branching. So both of these fibers will contract at the same time. All right, well, that's our coverage of microscopic skeletal anatomy. We'll look forward to covering this more in lab this week. But before we go, I just want to cover what's coming up in lab. We're getting near the end of the semester here. So just a reminder, enjoy Thanksgiving break next week. There will be no labs, so you'll get the week off. When we come back, so December 2nd through the 4th, depending on which day your lab is, we're going to have a review lab. We're still meeting. You still have to come to lab. There will not be any new material, but it's a chance for you to come, for you to look at the lab materials, review and study any of the topics we've covered since the last exam. So it's a nice chance to refresh, a good chance to go over some of those muscles you may have that may have gotten a little hazy in the last few weeks, and give you a last chance to ask any questions and do that prep in the labs. Then the final week of classes, we'll have our second practical exam. As a reminder, worth 25% of your grade. That'll be during our regular lab meeting of the last week of classes. So we will not have any exams during finals week for the lab portion of this class. So December 9th through December 11th, we'll be holding those exams. And at that point, you'll be free to study for the rest of your classes during finals week. Thank you. We'll see you in lab this week.